Howdy, today we'll be talking about Chapter 4, Methods to Study Viruses. So the first thing we're going to be talking about is growing these viruses. So the requirements for culturing cells and tissues are sterile conditions. The process is, first of all, we will mince the glands of our tissue or animal culture. Then we will add the enzyme collagenase. Finally, we will filter and grab the pellet. There's also an option to further separate these cells with differential centrifugation. There are the differences between the cultures of primary cells versus transformed cells. These are propagated and limited number of passages before it dies, so very limited with these, and these are the ideal ones that virologists use because they can be passaged indefinitely, so they're basically immortalized. Finally, second of all, we're going to be talking about purifying the viruses. So centrifugation, it separates macromolecules by density in artificial gravitational fields. So this image, that's what it's showing over here, how the density of all these particles gets separated after centrifugation. Next, we're going to visualize the viruses. So electron microscopy allowed individual viruses to be seen for the first time. There's negative staining, electron microscopy, thin sectioning EM, and cryo-EM. So for negative staining EM, it is a poor internal visual. It reveals the surface by using heavy metal coat surfaces to make a cast. Thin sectioning reveals internal structures and is fixed and in resin. Cryo, there is no need to be fixed or chemically stained and shows viruses in their natural aqueous environment. Fourth of all, we're going to be counting viruses. So there are two ways we can do chemical physical methods and infectivity titter, which only counts infectious viruses. So we have the visualization by EM, as we spoke to here before. We have hemagulation assay, which the virus is attached to RBCs in order to count them. Then we have serological assays like ELISA. So this one is an antigen plus a primary antibody. These bind together, they wash them, add a secondary antibody that can be detected, and finally they can detect the number of them. Fourth of all, we have genome quantification by PCR. So this detects viral nucleic acids, genomes, and transcripts. So next, we also have the infectivity titer. So we use the plaque assay in this case. These count viruses that lyse or kill host cells, so only infectious viruses. One plaque equals one descendant of infectious particle. And here we have a little image talking about how the plaque is visualized in a petri dish. Next, we have serological techniques. So we have the hemagulation inhibition assay, which detects antibody to virus or identifies suspect virus. Second, we have virus neutralization assay, used with plaque assay, detects antibody that can inhibit virus replication. So we have the ELISA that we just talked about previously, and then we have cell-based fluorescent antibody assay, where specific antibodies are tagged with the fluorescent molecule to detect viral antigens in the cell. Fifth, we have immunohistochemistry, where tissue is incubated with enzyme-tagged antibodies. This allows the examination of individual virus-infected cells in tissue section. Sixth, we have Western blots. This is a very expensive assay. Then this confirms identity of a reactive antigen by electrophoresis of the antigen. Sixth, we have detecting nucleic acids with the PCR. As we mentioned above, it is used to identify and or quantitate viral genomes in a sample. And we have high throughput sequencing, which is unbiased sequencing of DNA and RNA. And finally, the basic principles of diagnostic virology. They are to identify agent most likely responsible for causing disease in human and animal patients. These are the ideal conditions for the assays. They must be sensitive, specific, and rapid. Thank you.